This topic is DNA content during cell cycle. Cell cycle is a series of events which helps in the division of mother cell to divide to form either two daughter cell in case of mitosis and four gametes in case of meiosis. So the amount of DNA changes from one stage to another stage and is very crucial in the normal cell division. The amount of DNA in the cell is expressed in the form of C in haploid cell. Since haploid cell have 23 chromosomes, either from the fathers or from the mothers. So therefore, the somatic cells have 2C because 23 pairs of chromosomes are there. And thus, C is a unit of DNA. So first of all, we have to know what is the DNA content during cell, so during interphase. Your interphase is the first stage of cell cycle. The interphase consists of gap 1 synthesis and G2 phase, and G0 phase is also the phase is a resting phase, quiescent stage, suspended stage, where some of the specialized cell which never undergo further division and enters the G0 phase as nerve cell, muscle cell, cardiac cell. So now in normal all cell contain G1, S and G2 phase. In G1 phase, the DNAs are chromatin fibers beads on a string in the interphase. So now G1 prepare all the necessary protein enzymes for duplication of the DNA. So here the DNA duplicate in the S phase to form two molecules of DNA attached with the centromere. Each chromosome duplicate to form two molecules but attached to the centromeres, and thus the DNA become double. And thus similarly the G2 phase has the same amount of DNA, same content of DNA, which helps to further undergo cell division. So graphically, if a y-axis determine the amount of DNA in C and this the different phases, so in G1 phase the DNA is 2C in the somatic cell, in S phase 4C, and in G2 phase it is 4C. Now how this G2 phase DNA enters a mitosis and meiosis and form different amount of DNA. So the DNA contained during mitosis. As the DNA of G2 is like this, the DNA is doubled, attached with the centromere, it enters a prophase, condense, and in a prophase, condense to form the chromosome. Now this chromosome will bind with the spindle fibers and thus at the equatorial region during metaphase. And now during anaphase, the splitting of the centromeres occurs. From the centromere, the chromatide sister chromatide breakage occurs and the sister chromatide passes to two opposite poles. And then the telophase, here again the chromosome recondense to form the spindle fibers, the chromatin fibers, and thus the interphase, again the interphase state occurs. This is the telophase, and after that the cytokinesis occurs where the mother cell divide to form two daughter cell. Now, so the DNA was doubled in G2 phase, which passes to the prophase. The DNA is same. DNA is same. Here also, though the chromatin fibers are divided to two opposite poles, the DNA remains the same because it is occurring in the same cell. Even the telophase, though two nuclei are there in the single cell, but is inside the single cell, the amount of DNA will be same. But only after cytokinesis, the amount of DNA get halved. So, what we find graphically that during G1 2C, the somatic cell, the DNA was 2C, S double 4C, G2 4C, and subsequently prophase, metaphase, anaphase, telophase or have 4C of chromosome. And thus after cytokinesis, when two daughter cells are formed, two daughter cells have equal amount of DNA. So 2C equal to 4C, and thus what we found initially, the 2C was present in the somatic cell. Now also the 2C is present in the daughter cell. Thus the chromosome number as well as the DNA content remains same. See, the chromosome was 2 chromosome in the somatic cell. Again we can find the 2 chromosomes in the daughter cell. Thus the chromosome remains same and also the DNA content also remains same. So this mitosis also helps to make the DNA content and the chromosome constant. Now what happened during meiosis? So meiosis, as we know, takes place in the germline cell and helps in the production of gamete. It's very crucial to find out how the DNA content changes because meiosis have meiosis I and meiosis II, and in between meiosis is the interkinesis, the phase between two meiosis. Now in meiosis I, suppose this is a G2 cell, it passes to prophase 1 In the prophase one is the longest phase of meiosis, where uh, chromosome pairing occurs, the crossing over occurs, which are discussed in the cell division top lecture. Now, this prophase 1 passes to metaphase 1, where the homologous chromosome get bind with the spindle fibers. And then in anaphase 1, now the homologous chromosome passes to two opposite pole. One chromosome of one homologous chromosome passes to opposite pole. So one move to one pole, another partner move to another pole. There is no splitting. There is no, what we found in the mitosis was splitting of the chromosome. Okay, the daughter cell is splitted, divided, but no division. The complete homologous chromosome passes to different pole, and thus, and after in telophase they form the nuclei in the same cell, and after cytokinesis is formed the two cell containing one chromosome each. So initially the cell kept have two chromosomes, but now in the daughter cell one chromosome each, but the chromosomes are in the duplicated forms, not a single double-stranded DNA, but two molecules attached with a centromere. But the chromosomes become half. Now, so it was of meiosis 1. Now, what is the need for meiosis 2? You will come to know later. So, suppose if you see this cell undergo meiosis 2 to become prophase, the chromatin fibers condense to form the chromosome, now attached with the spindle fibers at metaphase, and then anaphase is similar to mitosis. Meiosis 2 is similar to mitosis, where the splitting of the centromeres occurs and helps to divide the sister chromatide to two opposite poles. Now you can find the DNA content which are doubled in this chromosome become half. Only for this reason the meiosis 2 was much important. So the anaphase 2, the sister chromatide moved to two opposite pole, and then here what happened? At telophase, the nuclei form in the same cell, and cytokinesis, the two daughter cells are formed. Here the DNA content is the DNA content becomes C, okay, and also the chromosome is also N haploid. Now what happened to this sister daughter cell is also undergo the same process. The prophase 2, metaphase 2, anaphase 2 with the spindle centromere breakage occurs and sister chromatide move to two opposite pole, and then it forms the telophase 2 where the nuclei form, and then cytokinesis. So 1, 2, 3, 4. One mother cell divide to form 4 daughter cell. Now graphically, what is the amount of DNA? So during interphase, the chromosome from 2C duplicate to form 4C. It will be 4C. 4C. Now this 4C chromosome progress to prophase 1, metaphase 1, anaphase 1 and telophase 1. And cytokinesis 1, the first meiosis end up when the cytokinesis occurs. So two daughter cell have 2C chromosome. See, the chromosome is 2C. Okay, the chromosome is 2C because each chromosome is double but attached with the centromere. Now, so meiosis 2 is important because this 2C, the cell containing 2C chromosome will now progress, okay, prophase 2, metaphase 2, anaphase 2, telophase 2, and then after cytokinesis become 1C because this sister chromatide separate splitting of centromere occurs. So 1C, 1C. So we get 1C chromosome. But in mitosis, we found the daughter cell having the 2C because the somatic cells have the 2C. One pair from the father and one from the mother. And now the DNA is 1C. Okay. So what was the need for this one? So meiosis, one mother cell divided to form 4 daughter cell. So what was the need for this meiosis 2? Because suppose when syngamy occur, when fertilization occurs, so one male gamete, suppose this is male gamete, okay, this is sperm fertilized with the egg, the female gamete. So the chromosome is N and DNA is C, 1C. Chromosome is N, DNA is C. So when fertilization occurs, this fuse to form zygote having 2N diploid and 2C. So see, the somatic cell is now same. So meiosis 2 helps to keep the chromosome number as well as the DNA content same.